grade 8 students, and welcome back to the biology class. At the end of this video, you will be able to relate a graft rejection to cell-mediated specific immune response, define non-self, and give examples. At the beginning, let us recall together the most important information that was explained in the previous video. We state that a graft is the transfer of tissue or organ from one individual to another, and this occurs in isograft and allograft. Also, graft can occur by transfer of tissue from one side to another in the same individual, and in this case we talk about autograft. Moreover, we state that all the nucleated cells, mean the cell with the nucleus, express, mean shows on its membrane a specific protein molecules. These protein molecules will be unique for each individual, and these specific protein molecules are called H.L.A molecules. The set of H.L.A molecules in all the nucleated cells in the same individual will be recognized as self. We state also that H.L.A play an important role in graft rejection, where when the H.L.A molecules between the donor and recipient are low similarity or different, the graft will be rejected. Remember again, rejected means not accepted. Example here, we have the H.L.A molecules of the donor and that of the recipient are totally different. That's why the graft will be rejected. And in the case of a graft rejection, the organ that was transplanted will be destroyed and no more functions. The question raised now, why a graft is rejected when H.L.A molecules are different? How does the immune system recognize the grafted organ and how it will act against it? To get the answer of these two questions, please concentrate and follow us. We stated that all the normal cells in our body are described as self. But when we say that the cell will be recognized as non-self, in fact, when the normal cell becomes an infected cell, it will undergo a change in its identity. The change can be occur in its shape and also in the H.L.A molecules. Example here, this is a normal body cell expressed on its membrane the specific H.L.A molecules. Once it becomes infected body cell, it will have a change in the identity of the H.L.A molecules. Observe it clearly, please, in order to guess the difference. Great. If you observe, this H.L.A molecules become a triangular shape. This one become round shape. Moreover, we have a new H.L.A molecules to be present where it's not expressed in the normal body cell. Due to this change in the H.L.A molecules, once the cell become infected cell, so the body will recognize it as non-self. Moreover, the normal cell in the body can undergo harmful changes where it transforms into cancerous cell. Example, the liver cell. If you observe it clearly, the normal liver cell and the cancerous liver cell, you will be able to notice that there is a change in the outer shape of the cell. Moreover, also in the change in the shape of the nucleus of the liver cell. So simply, once the cell becomes cancerous cell, change in its shape and the nucleus will be present, and that's why the cancerous cell is recognized as non-self. We state that each individual is unique and has his own biological identity, which is represented by marker molecules H.L.A that is recognized as self. In case of a graft, once the donor is different than the recipient, we look directly to the similarity of the H.L.A molecules between the two. Thus, the H.L.A of the recipient, of course, will be recognized as self. However, the H.L.A molecules of the donor because they are all totally different than that of recipient, will be recognized as non-self. And that's why the immune system will recognize the graft as foreign and will destroy it. Now, of course, you are asking yourself, how does the immune system act against the grafted organ? What are the cells involved in the case of graft rejection? That's what we are going now to explain it in this part of the video. We stated that once T helper cell bind to the macrophage, it will be activated. This activated T helper cell will secrete chemical. These chemicals will activate other types of lymphocytes such as T killer cell or B lymphocytes. Remember again, in some cases it will activate both of them, T killer cell and also B lymphocytes. But which of these two cells do you think will be responsible to play an important role in graft rejection? That's what you are going to notice it in the next slide. We stated that in case of a graft rejection, the immune system recognizes the grafted organ as non-self, mean foreign body. That's why the immune system is going to fight it, while T helper cell is constantly scanning the cells and the particles in the body in order to keep it safe and to detect any foreign, exactly like the police in the roads. So, the T helper cell will bind to the grafted cells and recognize the HLA molecules and identifies it as non-self. Thus, it will turn on the alarm. The T helper cell now will alert the T killer cell, by which in turn it will be responsible to act against the grafted organ, which is recognized as non-self, by secreting chemicals that cause the explosion for the cell where it destroys it, mean it causes for the death of this non-self cell. Now, we are able to state that the type of the immune response involved in the graft rejection is cell-mediated specific immune response, where the T killer cell is responsible to be active. As an application now, Ali had a skin burn and wanted to have a skin graft. Classify among the following the cells considered as self or non-self with respect to Ali. Please pause the video and write the answer on a draft paper. Let us start with the first one, Ali's brother cell. Brother, of course, it will be a different individual from Ali. Thus, the H.L.A molecules are totally different, so it will be recognized as non-self. Ali's neighbor's skin, it is similar to the first case, so also will be recognized as non-self. And the third one, a piece of skin from Ali. So we are talking in the same individual, mean identical H.L.A molecules, so of course it will be recognized as self. Ali's infected cells. Remember, infected cells, it will be recognized as non-self, where it will be totally different from the normal cell concerning the shape and also the HLA molecules. Ali's identical twin skin, since we are talking about identical twins, thus identical HLA molecules, thus of course it will be recognized as self. As a summary, what can we say about graft rejection? We say that the grafted cells carry different HLA molecules than the recipient HLA molecules. Like here, the HLA molecules of the recipient is totally different than that of the donor. And that's why the immune system will recognize the grafted cell as non-self. Because it will be recognized as non-self, so the immune system will act against the foreign body and destroy it. How? By the action of the T helper cell, which recognizes the graft as foreign by binding to its HLA molecules, and it secretes chemicals to alert T killer cell. The T killer cell in turn binds to the grafted cell and secretes chemicals that causes the explosion of the grafted cell and thus its death. At the end, we can say that the graft is rejected. We recall again, since the T killer cell is activated in the graft rejection, so the immune response involved in the graft rejection is cell-mediated specific immune response. At the end, I want to thank you, dear students, for your attention.